Hi, everybody, and welcome to our last lecture in this series. And what we're going to be looking at is the final step, which is going to be the electron transport chain, also known as the ETC. Now, what we're going to be learning is where does this happen? What are these specific steps when it comes to electron transport chain? And then what do we make, or what is our end goal? What are we trying to produce from this portion? So looking at the electron transport chain, there's a couple important things we need to identify when it comes to this. Now, the electron transport chain is essentially a series of protein complexes in the mitochondria that pass electrons from one of these complexes to the next, finally to oxygen. So the electrons are passed to the chain from NADH and FADH2. So remember, these were produced from our glycolysis in step one, as well as our Krebs cycle in step two. So as the electrons move through the electron transport chain, the electrons lose energy, and that energy is used to pump hydrogen ions across an inner mitochondrial membrane, creating an electrochemical gradient. So remember, when we're dealing with anything with diffusion or gradients, the molecules always want to move from high to low concentration. So what's going to happen is our H plus is going to diffuse down the gradient through something known as an ATP synthase, which is going to act as a tiny generator turned by the force of the hydrogen ions diffusing through it. This turning of the molecular machine is going to generate our ATP from ADP. So looking at that, that was a lot. To kind of sum things up, in our electron transport chain, we have a lot of series of protein complexes. And what's going to happen is this is going to be in the mitochondria, and it's going to be passing electrons from one of those complexes to the next. What we're going to do in order to kind of help facilitate the electrons passing the chain, we're going to utilize NADH and FADH2, which again, we were generated from our glycolysis as well as our Krebs cycle. So again, we went through our first step in order to produce some precursors for this. We did produce a little bit of ATP, but our main step was to produce those precursors. Same thing with the Krebs cycle. We wanted to make those precursors for this specific portion. And the reason why I keep stressing that is because the electron transport chain is going to produce a massive amount of ATP. It's going to do that using our hydrogen ions across our electrochemical gradient. Again, remember, we're moving from high to low, so that's why we're pumping those. And again, that H is going to diffuse through that gradient, and it's going to go through something known as our ATP synthase, which is going to be our tiny generator in order to produce ATP from ADP. Remember, ADP is going to be very low in energy versus ATP is going to be very energy dense. So let's go ahead and see exactly what that looks like and kind of what's going on. So looking at this over here, remember, it's going to happen in the mitochondria. We went through the glycolysis, which was in the cytoplasm. That moved over to the Krebs cycle. Remember that cycle has a lot of biochemical things happening here, and it's going to continuously be reusing all those things in order to produce precursors, again, for the electron transport chain. So in this case, what we're doing is we're taking electrons from NADH and FADH2, which you can see right here. Again, we produce those in our last steps. And what's happening here is, remember, they're going to be grabbing those electrons, and those electrons are going into, remember, those protein complexes in our electron transport chain. Now, the reason why it's known as the ETC is you can see each protein complex is going to be passing along that electron from one to another. And then finally, we're going to end up with a byproduct of oxygen, and we're also going to have our water. So in this case, again, we're taking in the electrons from the NADH and FADH2, and what we're trying to do is output or produce ATP. We're trying to produce roughly around 34 to 36. So this is going to be considerably much larger amount than the other two cycles. Again, ATP is our energy dense molecule that we want to produce, and we're going to be doing that by, again, every time we're passing these electrons out over, we're pumping in our hydrogens. So now we have a very high concentration of hydrogen up top, and we have a very low concentration on the bottom. And remember, things want to be in equilibrium. So what's going to happen here is we want to equal this out. So we have that high concentration, and it's going to go through this ATP synthase. This ATP synthase is going to essentially spin. As the hydrogens are moving down, it's going to start taking a D piece. Remember, adenosine diphosphate, we only have 2 phosphates here, and it's going to go ahead and add that additional phosphate in order to produce our ATP, or adenosine triphosphate, which is going to be 3. So let's go ahead and take a look at what those steps are going to look like in actual writing. So in step 1, we have our NADH and FADH molecules, remember, generated from both our first step, glycolysis, and our second step in the Krebs cycle. And they're going to transfer high energy electrons to the electron transport chain. In step 2, as the electrons are passing along the electron transport chain, hydrogen molecules are going to be pumped across the mitochondrial membrane to form the hydrogen concentration gradient. Remember, going back here, you can see we're pumping that in to produce a higher level of hydrogen concentration here and creating a lower concentration below. So oxygen is the final acceptor for these electrons. This requirement for oxygen is why you need to breathe for that O2. So again, you can see we have our final acceptor here, and then we're going to end up with our byproduct of H2O. So hydrogen moves down that concentration gradient through the ATP synthase, which is going to use that movement of that hydrogen to produce roughly around 36 ATP. So again, this hydrogen over here is going to allow that spinning and production of the ATP synthase, again, taking ADP to produce ATP. So again, this is what that's going to kind of look like. You can see step 1, electron transport chain. The electrons transfer to the electron transport chain here. Again, we're taking these electrons and we're putting them into these complexes. Number 2, hydrogen pumps across the membrane, creating a hydrogen concentration gradient. Again, you can see these hydrogens, again, are being pumped up. And the reason why they're being pumped up is due to the electrons. Again, they're transporting each other in order to activate these to pump in that hydrogen. We're creating a high concentration now of hydrogen, and now we have a low concentration here. And because of that concentration difference, or that gradient, high is going to want to move to low. So after all of these electrons, again, are going to go through this chain down here, oxygen is going to be that final acceptor right here for the electron. So again, that's why we need to breathe O2. So we need oxygen. It is going to be a requirement for the electron transport chain. So once we've done that, remember, we still have that concentration gradient, high hydrogen here, low over here. So we want to equal that out. So hydrogen is going to diffuse through ATP synthase, this piece right here. And as it's moving through, it's going to start to spin and generate, again, taking in ADP to produce ATP. So again, ADP, we only have 2 phosphates, and then we're adding another one, so we end up with 3, adenosine triphosphate. So again, that is going to be the steps to the electron transport chain. So in summary, for cellular respiration, we have 3 steps. We have our glycolysis, which is going to be in step 1. And all we're trying to do here is we're trying to go ahead and break apart the glucose. Remember, we're taking this in, and we're going to go ahead and separate that out to produce pyruvates. Those pyruvates are also going to have our 2 ATP and our 2 NADH. Remember, those electron carriers. So this right here is a precursor to the electron transport chain. Now, once we have these pyruvates, 1 or 2 things can happen. If we have oxygen, right, O2, it's going to go to the Krebs cycle. If we do not have oxygen, it's going to go through anaerobic respiration. Again, this is going to be triggered because of low oxygen levels in red blood cells. If this is the case, we end up going through fermentation and producing lactic acid. So in this case, we have a low amount of ATP. But again, when we have oxygen, we're going to go through the Krebs cycle. In the Krebs cycle, we're going to go ahead and spin through that biochemical cycles in order to produce ATP here. Again, we do have some CO2 that's going to be a waste product, but the main thing that we're trying to produce is going to be this over here, our 6 NADH and our 2 FADH2s, which again are going to be those electron carriers. So again, we produced this NADH from our glycolysis, and then we went ahead and produced our NADH here, 6 of those, as well as 2 FADH2s, again, all being electron carriers. So we're going to finally go ahead and go into the electron transport chain. Remember, all of these are going to do, they're going to go ahead and drop off all those electrons, and they're going to go ahead and go through those complex pumps. And the reason why is because we're trying to push hydrogen into the top, creating a gradient. So we have an area of high and an area of low now. And the whole reason why we do that is because we're trying to spin our ATP synthase. It's going to spin down here, and we're going to go ahead and produce ATP. Okay, again, we're taking a DP, adenosine diphosphate, which is very low in energy, and we're going to go ahead and produce a TP, in this case 36. So this is why the mitochondria is commonly known as the powerhouse of the cell, is because it goes through these 1, 2, 3 cycles in order to produce our ATP. All right, with that being said, that is going to conclude all of those steps for cellular respiration. So there are going to be a bunch of questions, practice problems for you. So what I'm going to do is I'm going to go ahead and give everybody a couple minutes, or I'm sorry, a couple seconds to give this a try, and then we'll go and look at the answers, and then the same thing for the subsequent slides. All right, and this is the first set of answers. And then this is the second questions. There goes the second set. And then these are going to be slightly different. So you have a diagram here. What you're going to do is give A, B, and C a try. Again, these are all going to be questions that have to do with the electron transfer chain and what's the issue with this. Essentially, something's going to happen. There's going to be a lack of something, and then what essentially is being affected. So same thing, I'm going to give you guys 5 seconds to give this a try. Okay, and then this is that last type of question. Same thing, give everybody about 5 seconds to pause and give this a try. All right, and that is going to conclude our lecture. So again, as always, if you do have any questions, comments, concerns, please feel free to reach out, and I'll see you all in the next lecture.